We're going to talk now about what's called the membrane potential and how it accounts for the excitability of cells. We're going to see that ion flow is responsible for maintaining the resting potential across cell membranes. The resting potential is a voltage difference across cell membrane. You may recall that cells are slightly electronegative in the cytoplasm relative to the exterior of the cell. Cells are irritable. That's one of the major properties of life. That's because they respond to stimuli, and the stimuli that they respond to disturb the resting potential. We're going to see then that ion flows not only account for maintaining the resting potential, but are responsible for the irritability of cells, the actual ability of cells to respond to stimulus. In this cartoon we have a voltmeter with one of its electrodes penetrating the membrane of a cell. And this is how we would measure the resting potential. The voltmeter will register that minus 20 to minus 200 millivolts, telling us that the interior of the cell is indeed negative relative to the outside of the cell. The resting potential ranges from about minus 20 to minus 200 millivolts. This will depend on cell type. Most cells have a resting potential around minus 70 to minus 80 millivolts. Cells respond to stimulus with local disturbances in that resting potential, and neurotransmission is an example of this. Let's back that up one time and look at it again. So when the nerve cell or part of the nerve cell appears, the arrow is the propagation of a nervous impulse reaching the nerve ending, and as the impulse reaches the nerve ending, it causes a release of neurotransmitters which crosses the synaptic cleft. The asterisks in this case are the neurotransmitters. Let's look at that again. And they are released from synaptic vesicles. They cross the cleft, and when they bind to their receptors in the responding cell, it induces a localized membrane depolarization, and at that little location, right where the nerve cell has innervated the responding cell, the polarity of the cell actually switches, and now the interior of the cell is positive with respect to the outside. Now that doesn't occur around the entire cell, but it's a localized membrane depolarization. That's how nerve cells stimulate responding cells. Could be other nerve cells that are responding, or also muscle cells will respond in much the same way. Well, I said that ion flow is responsible for maintaining membrane resting potential and for response to stimulus. Well, ions, of course, are highly charged, and they cannot cross the phospholipid bilayer by themselves, so they cross through ion channels. We recognize two kinds of ion channels. Channels which are, for some ions, almost always open, or they seem to be open all the time, and others that are regulated, that is, can be opened or closed in response to a certain stimulus. And these are called gated channels. Let's take a look at what causes a gated channel to open and allow the flow of ion. One thing could be a change in membrane potential. I didn't discuss it in the last slide, but the neural impulse that is propagated down an axon is a little voltage jolt, and when it reached the nerve ending, it actually opened a voltage-gated channel in the nerve ending membrane. Now we're going to look at that in some detail later. But that would be an example of a voltage-gated channel. We also saw in the cartoon of how a nerve functions the binding of a chemical, the neurotransmitter, to the responding cell, and we're going to see that that binding opens another ion channel, this time in the responding cell, and therefore that channel is considered to be chemically gated or ligand gated. Ligand is just a fancy word for the kind of molecule that binds to the channel protein. Okay, so that's a ligand gated channel. There are also mechanical stress stimuli that regulate mechanically gated channels. We won't have a chance to look at those, but you can read about them in the textbook. There's a neat example of one there. Needless to say, these channels are proteins. They are proteins that can respond to a voltage jolt or to the binding of a chemical like a neurotransmitter, or in the case of stress, they can actually be slightly deformed by a stressor. Let's look at the patch clamp recording technique for actually measuring ion flow. Inside a micropipette that you can actually see in the photograph in the upper right of the patch clamp device, there is a solution that can detect the presence of ions. And these solutions can be modulated or varied, composed in different ways to recognize different ions. And so we can recognize specifically, in this case, potassium. We could design a patch clamp device with a sodium ion detector or a chloride ion detector, so we can detect cations as well as anions. And this is a picture of a patch clamp device. The pipette is sort of sucking at the cell, which is at the left, creating a little protuberance that protrudes into the pipette of the patch clamp and creates a watertight seal. And it is possible to have a voltmeter, again, built in such a way that the electrodes travel through this patch clamp pipette, and one electrode, again, can penetrate into the cell. And so you can measure the flow of ions as a voltage change and actually detect specific ions. We have drawn here in green a channel of some kind. So it turns out that the resting potential of cells is due to a kind of general leakage of potassium out of the cell through potassium channels that are always open. Potassium is higher in concentration in cells than out. So if the potassium ions can get out of the cell, they're going to leak out of the cell. They cannot cross the phospholipid bilayer. So if they're going to leak through some kind of channel that allows them to pass through. And you can detect that with this patch clamp recording device. You can also introduce a power supply with electrodes that penetrate the cell so that you can give the cell a little jolt that will cause a local depolarization. If you depolarize the cell, you can also measure ion flow, and if you measure ion flow in response to a synthetically generated electrical potential difference, you are looking at a voltage-gated ion channel. Okay. I said that the resting potential itself results in part from potassium flow through leak channels that are always open and allow potassium just to flow out of the cell. So I said that potassium is at higher concentration in the cell than out. Wouldn't you expect that the potassium would simply flow out of the cell until the concentration of potassium in and out of the cell is equal? In other words, until equilibrium is reached. That doesn't happen. So yes, potassium is higher inside the cell than out, and potassium does leak through these leak channels, but they only do so until an electrochemical gradient prevents further potassium leakage. How does that happen? The negative charge inside the cell, the negative ions, are mostly organic ions, and these are mostly organic acids, which have, say, carboxyl groups, which dissociate to COO-, or phosphates, which at the pH in a cell, which is around 7, also exist as negatively charged ionic groups on molecules. So those organic anions are the source of most of the negative charge in a cell. So the cell has negative charges, and if the positive charges that would otherwise neutralize these ions were to leak, eventually you'd get an electrochemical gradient, wouldn't you? The cell would in fact become electronegative relative to the outside. And then the potassium ions, which are positively charged, would slow down. They wouldn't leak as fast, and eventually they would stop leaking altogether. So the potassium leakage stops when the cytoplasm cannot be made more electronegative. That is, there is a restraint on the leakage of the potassium. And that is largely what establishes the resting potential. It is established by the balance between the tendency of potassium at high concentration to leak out of the cell through these leak channels and the tendency of a growing negatively charged interior of the cell to require positively charged ions to neutralize them. We're going to see now how ion flow results in a stimulus of a cell, in the disturbance of the resting potential. So membrane depolarization occurs when cells are stimulated. In other words, the resting potential is disturbed. Stimulation of a nerve by a nerve or a muscle by a nerve generates what's called an action potential, which looks like this. So if you have a voltmeter with a one probe, one electrode in the cell, one out, and you stimulate the cell at the point shown here in the graph, you can watch the voltage change. And the voltage goes, in this case, from -70 to about +30 or +40 millivolts. And then the resting potential is reestablished. That rise, depolarization, in fact a hyperpolarization, as the polarity of the cell not only goes to 0, but exceeds 0, so in fact the charge is now positive inside the cell compared to outside instead of the usual resting potential. That's called hyperpolarization. It reaches a peak and then comes down. So we can look at it again. And you said it's very fast, so it goes up and then it goes down. It happens on the order of milliseconds. We'll talk more about the action potential and its relation to ion flow in just a moment. But first, let's look at how these gated channels work, or at least what they do in some minimal detail. So this is from your textbook. Gated channels can be either open or closed, and this is really the take-home message of this slide. They can be open or closed, and they will regulate ion flow to allow, as it says, response to stimulus. We have a voltage-gated channel on the left. A depolarization will cause the channel to open, and ions will flow in the direction from high concentration to low concentration. That part is not mysterious. Item B is ligand-gated, so this could be the acetylcholine receptor of a muscle cell, for example, responding to acetylcholine released by a neuron that is innervating that muscle. So the little upside-down red teardrops might be acetylcholine binding to a receptor site, and the result is to open this channel to allow ions to flow and for a response in the muscle or next nerve cell. We have ligand-gated intracellular and extracellular channels. We won't talk too much about those. And there's an example of a mechanically gated ion channel. Go to the textbook to check that out. So we're going to look at nerve cell function as an example of how ion flow disturbs resting potentials and how that disturbance results in a response. So let's dissect the action potential I drew a moment ago into ion flow events. Action potentials are generated when sodium ions flow into cells. So earlier I said that potassium ions are at higher concentration inside a cell than outside a cell. It turns out the sodium concentration is low inside the cell relative to outside the cell. So sodium concentration will always be somewhat higher outside the cell. So here are our axes, the x-axis being time and the y-axis being membrane potential in millivolts. We have a voltage-gated channel, which is in its closed state to start with. So ions cannot flow through this voltage-gated channel. If the time of stimulation is at the point of the arrow shown here, after stimulation, the voltage-gated channel opens and sodium rushes into the cell. The rushing in of sodium, those positively charged ions, reduces the negative charge inside the cell and in fact creates a localized positive charge inside the cell, a hyperpolarization if you will. And when that hyperpolarization reaches its peak somewhere around 35 or 40 millivolt positive, the voltage-gated channel is inactivated. It's being capped, if you will. It's being shut off. So there's no further ion flow, so no further sodium can rush into this cell, and therefore the peak of depolarization can't get any higher. That then allows the cell to return to the resting potential, which we see here. Now, the return to the resting potential occurs over time, of course, and is assisted by a voltage-gated channel that has now opened to allow potassium flow, i.e. potassium leaves the cell. So what's happened here? Let's go back really quickly. We have a voltage-gated channel that is in its closed state until stimulation, and when stimulated, the channel opens and sodium rushes into the cell. When sodium has rushed in sufficiently, the channel is inactivated. There's no further ion flow, and a voltage-gated channel opens. This channel lets potassium leave the cell so that some of the positive charges that rushed in the form of sodium ions can at least be reduced by the exit of potassium ions. I'll point out that the voltage-gated potassium channel was activated by the membrane depolarization that occurred in the first place. So we have a voltage-gated potassium channel opening up and allowing potassium to leave the cell. This results in a return to the resting potential, at which point the sodium channel is now no longer inactive, but it is closed. So what we've seen here in looking at the sodium channel is that it can exist in three conformational states: closed before stimulation, open upon stimulation, and inactivated upon the maximal stimulation. And then eventually, in order that the cell can respond again and generate a second action potential, the resting potential has to be reestablished and this sodium channel has to be closed but not inactive, has to be in its original conformation. I mentioned that an action potential is a local event on a membrane. That is to say, the cell doesn't instantly become positive on the inside relative to a negative exterior when stimulated. This depolarization and even hyperpolarization occurs only at the point of stimulation, say by a nerve cell, or if you're doing it with a power supply artificially, just at the location where you applied this jolt of electricity. So it's a local phenomenon. Having said that, however, that local depolarization or hyperpolarization can be propagated. So the nerve cell we saw before, we saw a cartoon of a part of a nerve cell, it didn't show you what was happening at the other end of that axon, in fact, in what's called the cell body of the cell, which probably received a signal from another nerve cell, a depolarization signal, which was then propagated along the axon until it reached the nerve ending. So here we have a cartoon based on something in your text of a region of the axon. And if a depolarization were to be propagated, that means it's traveling from one end of the axon to the other, to the nerve ending. And so we see here on the left a depolarization, which is being propagated along the axon eventually to reach the other end. And let's just look at this one more time. It's being propagated as these ion channels open and close and inactivate, open and close and inactivate in sequence. And that's how an action potential can be propagated once it's produced at one single point on a cell, propagated along the membrane, along the axon in this case, until it reaches the nerve ending, where that electrical impulse stimulates voltage-gated channels to do their thing. So these are voltage-gated channels that lie along the axon, and they respond to traveling potential difference by cycling through those three conformational states I pointed out before. And they do this, again, until that hyperpolarized signal reaches the synapse at the nerve ending. So now let's take a look at a nerve ending a little bit more closely and see how the ion flows are regulated. So here's our nerve impulse. It's moving along the axon, being propagated the way the previous slide suggested, until that potential difference reaches a channel protein, which in this case is a calcium channel protein. It's a voltage-gated calcium channel that opens up in response to that little electrical jolt that was propagated down the axon. The calcium enters the cell, interacts with synaptic vesicles, causing them exocytotically to fuse with the membrane at the nerve ending, and the neurotransmitters are released into the synaptic cleft, that is the space between the nerve ending and the membrane of the responding cell, and the neurotransmitters then bind to receptors on the responding cell. The response is that a ligand-gated sodium channel opens, beginning an action potential. So what we've seen here in nerve action is the propagation of a nerve impulse that is mediated by changing conformation of ion channels all along the axon, resulting in that nerve impulse, which could be as much as a foot or two away. You know, some of these nerves have very, very long axons, and that voltage difference, that change in potential of the membrane, reaches the nerve ending and has these consequences. First, activating a voltage-gated calcium channel, allowing calcium in. The calcium, by the way, is a positively charged ion, which will activate voltage-gated channels that cause synaptic vesicles containing the neurotransmitter to actually fuse with the membrane. That's a bit that I left out before. And then the neurotransmitter itself is a ligand binding to a responding cell neurotransmitter receptor, which is in fact the sodium channel that opens to allow sodium to rush into the responding cell and initiate the action potential. So to summarize, the firing of a neuron, or for that matter a muscle cell, or any cell that is responding to a nerve innervation, causes ion fluxes. Sodium enters the cell, that leads to depolarization, that leads to the beginning of an action potential. Potassium leaves the cell, restoring the cell's membrane potential. And a cell can be excited several times like this, and each time it's excited, the resulting ion flows will cause sodium, which is at higher concentration outside the cell, to rush in, and to complete the action potential, potassium will then rush out, both events being mediated by these voltage-gated channels. But there's a problem. You can restore the resting potential, which will allow the cell to fire again. But at some point, you will have put so much sodium in the cell and lost so much potassium from the cell that the cell will no longer be able to fire properly. It no longer has the correct sodium-potassium balance. Restoring the sodium-potassium balance is the job of the sodium-potassium pump, an example of active transport. So we are looking at an active transport protein in the membrane. The sodium-potassium pump is highly understood. It's a well-studied example of active transport, and free energy is used to establish the gradient in which potassium then is again high inside the cell, low outside the cell, and sodium is once again low inside the cell and high outside the cell. We've seen an active transport system before. The mitochondrial proton pump is such an active transport system. It uses energy from energetically favorable electron transfers to concentrate protons outside the matrix in the intermembrane space, or even in the cytoplasm, since the outer membrane is leaky to protons, right? You remember that. The sodium-potassium pump gets its free energy from ATP, and it uses that ATP to reestablish those correct sodium and potassium gradients across cell membrane. So we start with ATP in the cell. The cell is off to the left. We have a sodium-potassium pump protein in blue, shown here. And we have sodium shown as purple triangles and potassium shown as brown spheres or brown circles. So how does the pump work? Well, the first thing that happens is ATP is hydrolyzed to ADP, and the phosphate from the ATP binds to the sodium-potassium pump. Let's look at that again. Because when the phosphate is used to phosphorylate this protein, let's look at it again. Watch the change in shape. Binding of phosphate results in a conformational change in the sodium-potassium pump protein, and in that configuration, the protein is able to recognize sodium molecules and expel them to the outside. We can look at that again. There are some purple triangles going to bind to binding sites. 3 of them actually bind, and 3 of them then are expelled. You'll notice that in the expulsion of the sodium ions, the pump has again changed conformation, and it actually has exposed some surfaces to the outside of the cell which recognize not 3 but 2 potassium ions. And in the process, the sodium-potassium pump changes conformation one more time and releases the potassium inside the cell. And we can look at that again. So here we have the potassium's binding, a very rapid change in configuration, and the potassium leaves the pump protein and is now inside the cell. And you'll also notice that the phosphate has come off. Part of changing conformation in this part of the illustration is that the phosphate is hydrolyzed off of the protein. So to summarize, free energy, G, remember, from ATP hydrolysis is what's going to power this pump. The phosphate actually participates in the process by binding to the sodium-potassium pump protein, causing a conformational change in which the sodium can bind and then be expelled from the cell. And the phosphate then is hydrolyzed as the potassiums go into the cell, and again, cyclic conformational changes account for the binding and release of the different ions. So this process reestablishes the normal sodium and potassium concentrations in a resting cell. So a responsive cell has a resting potential and a resting sodium and potassium concentration, which is then affected when the cell has to respond to an innervation or other stimulus. The pump, by the way, and I think I mentioned this, engages in a 3 sodium for 2 potassium exchange. So potassium, 2 potassiums are brought into the cell for every 3 sodiums that are expelled from the cell, the process of reestablishing sodium-potassium balance. I remind you then that making gradients requires free energy, and gradients actually contain free energy that can be used to do things, as we saw in the case of the mitochondrion, and as you probably read in the case of the chloroplast as well. And that brings us to the end of this presentation.